Started. So unfortunately, as I mentioned, this program here cuts off the pictures because it resizes them. So if you notice, the anionic form is missing, but we can draw that in. But the important thing here is the fact that the amino acid backbone actually is a Zwitter ion. Zwitter ion, as we mentioned before, and so it can have multiple ionic states based off of the pH. Okay. And so what that means is that we can have it to where, here, let me just see if this works. Whoops, wrong one. What does that do? C alpha in H3 plus. And then I'm just going to call it R. So R is what changes. So at low enough pH, everything has its hydrogen on it, so it has an overall positive 1 charge. Now remember, some of the side chains on the amino acids themselves can also have a positive charge. But in the simplest amino acids, it would all have at least a positive 1 charge. As you increase the pH, then it's going to start to lose its hydrogens based off of the pKa's. And the first one it's going to lose is the carboxylic acid. So then the nitrogen will still be there with its, but it will now be a carboxylate.
You said as you increase
pH. No, as you increase the pH, it's going to change the overall charge on the ion based off of the pKa of each group. And so we'll talk a little bit about the pKa's of each group in a moment, but the first one will be the carboxylic acid loses its hydrogen. So overall, on this first one, we had a +1 charge at least. Once again, some of the R groups on them could also be positively charged, but for the most generic, like if we were looking at glycine or alanine, it's going to have an overall +1 charge. Then it's going to have a neutral charge because we have a positive on the nitrogen and negative on the oxygen. And if you keep going to high enough pH, and this is reversible, it's called titration, you're actually going to then, let's see, C alpha, you'll still have the negative charge on the carboxylate, but now the nitrogen has lost its extra hydrogen, so it's neutral. So overall, we now have a -1 charge. Okay, so that's what they're talking about. And so you can do what's a titration. You can actually put in an amino acid and slowly add in, for example, if you slowly add in hydroxides and increase the pH, you can see it change from one form to another. Or you can start at another pH and add in hydrogens in order to go downward, and you'll see this profile. Okay. So the average pKa for our carboxyl group is 2.9, okay? Which is actually a stronger acid than what you would expect it to be. Because if you think back to organic chemistry, the simplest carboxylic acid that had 2 carbons was acetic acid, vinegar, essentially. And it has a pKa 4.76. So the question we need to ask is, why is there such a change in the pKa? Okay? And the reason being, and I bet it doesn't, let me see if it shows. Yes. The reason being is that due to that nitrogen just being 1 carbon removed from the carboxylic acid, is it affects. So it does it through, remember, it's called the induction or inductive effect. And so it's going to pull some more of the electron density towards that nitrogen, and it makes it easier for that hydrogen to pop off. And so instead of being around almost 5, it's roughly right around 2. I don't expect you to memorize all of the pKa's. I want you to just know that in general, roughly around 2 to 3 is the pKa for that carboxylic acid group of the amino acids. Okay? And so this is just what we've been talking about here. This one is actually looking at a generic amino acid. And so what we can see here is at really low pH, it's got an overall +1 charge. Okay? as you add in hydroxide, when you add in a 1 mole of hydroxide, you're going to completely compete off that hydrogen there. So that's whenever you're going to get that Switter anion, so it's neutral. You add in a 2nd mole of the hydroxide, it's now competed off the 2nd hydrogen, the one off the amino group, and so it's overall -1. Let's see, is there a way to erase all of these? I forgot this one's got the laser pointer. Okay, so I wanted to point out a couple things here. So this is called the isoelectric point. That's the pH at which the overall molecule of the amino acid is neutral. So on the simplest amino acids like glycine and alanine, it's just going to be the average between the two other pKa's.
It's the point at which what? It's
The isoelectric point, which is called the pi, is the pH at which the overall charge of the molecule is neutral. So that's what they're showing right here. Now, if you go to low enough pH down here, just like we said before, overall it's positively charged. At a high enough pH, it's going to be negatively charged. But then, if you just think back to Gen Chem, we have these 2 spots in this one, which these are the individual pKa's for, one is the pKa for the carboxylic acid, the other one's the pKa for the nitrogen, the amino group. And so there, it gets a little bit more, not difficult, a little more tricky when you think about the overall charge of the beaker of amino acids. The lower one, the pKa1, so that means that half of the molecules are going to be neutral and half of the molecules are going to be +1. So that means overall the charge here is going to be +1/2. Okay. So same thing will happen up here. When you get to this pKa, since that's the titration point, half of the molecules are going to be completely negative, but half of them are still going to be neutral, so that's why overall it's going to have a -1/2 charge. So those are just inflection points if you think back to calculus. So I just want you to make sure that, and some of the suggested problems are about the titration curves, and they talk about it in your book. Make sure you know how to do that and what that means, because it's important, especially from medicinally. Whenever you can do the same thing with medicinal chemistry, because based off of the type of medicine it is, it's going to have different pKa's, and you can figure out the overall net charge. Right. So this is just to show here that the pi is, once again, that's just, I can't see it, but it's supposed to say the isoelectric point, pi. This is pH, which is, once again, the net charge is neutral. Okay. So this shows you, for the most simple ones like glycine or alanine, then you can just take it by doing the average of the 2 pKa's. Of course, not everything is that simple, since we also have amino acids that have multiple pKa's. All right, once again, do not memorize this. But if you look, you should see that roughly right around pH 2, all of the pKa's for the carboxylic acid is right around 2. Likewise, here if we look at the pKa's of all of the amino groups, they're roughly 9 to 10, give or take. Okay, and then this table gives you the PIs. Now, what I wanted to show you though is, if we look at the slide, is there are some of the amino acids that the R groups also have a pKa. And so that's what this table is showing you with what they call the pKa of the side chain. Some tables may even say pKa of the R group or something like that. So, and it should make sense. I mean, some of this should make sense. We have here the aspartic acid has one, and it's going to be acidic, right? Because it's a carboxylic acid. So it's got 2 carboxylic acid. It has the one from the backbone, and then it's got the one from the R group. So its overall pi is really low. And then glutamic acid, tyrosine, cysteine, those were all acidic. Those were ones that could lose their hydrogens. Cysteine forms a thiolate, and tyrosine forms a phenoxide. And then we have our three that are bases, the basic ones. And so they have pKa side chains that are higher pH than, and so it alters the overall pI. So for example, here histidine, you see the overall pI, instead of it being around 6 or so like glycine, it's more basic. It's 7.6. But once again, don't memorize that. Just want you to know that the one for the carboxylic acid portion of the backbone is roughly around 2, and the one for the amino acid portion of the backbone is roughly around 9 or 10. Otherwise, I would give it to you. Or I could give you the graph and ask you, so where are the, where's the PI, where are the inflection points, the PKAs, and I'd expect for you to work backwards. And once again, just practicing what's in your book and the suggested problems, or even in if you got the study manual, that would help. This is really what I wanted to focus on. Wow, that color does not come through. That's the invisible word is peptide bonds. And so, guess the word is peptide bonds. Okay, so polypeptide just means multiple peptides. And proteins, building block are amino acids, they come together, and the special bond there is called the peptide bond. And we're going to talk about the special properties with respect to it. Yeah, this is a real shame that this one did not come through, because I just wanted to show you, you can see the little cartoon portion, but I was going to show you the portion down below, which you could have in your notes. So this is just a cartoony way of showing it. We have one amino acid. Here's the second amino acid. In that color right there is the R group. It's just a hydrolysis or condensation reaction. A condensation reaction. So that's why water's getting kicked off, and you have what's called the peptide bond. It's an amide formation. So all of the peptide bonds in the backbone of the protein are due to amides. Okay, and so that's what that picture is showing. For those in their notes, it looks something like this. Woo! And away it goes. That is so cool. All right. So what we have here is we have and next chapter we're actually going to go into more detail about this. C alpha. I'm going to call it R1. So that would have been the carboxylic acid end of the first amino acid, but it's now attached to the next amino acid. So here's the nitrogen group. Was it C alpha?
So the whole oxygen is given off.
Because it's in water. The nitrogen loses hydrogens and the oxygen is gone, yeah, because it's a condensation reaction. So this is now it's an amide.
So now it's connected to the double bonded oxygen, the R group, and it has 2 H's. So they got rid of 1.
It would only have 1 H. For the nitrogen, it's only going to have 1 hydrogen.
Oh, because an amide only has 2.
Right. And so yes, it just has 1 hydrogen. And like I said, next chapter we actually go into much greater detail. Now what they do is they give it a directionality. So this side right here is called the N terminus or the amino terminus. And so therefore that side would be called the C terminus or the carboxy terminus. All right. And so once again, that's just a simple peptide. If you have 2 peptides that come together, it's called the dipeptide. 3 means tri, 4 would be tetra. But then what happens is at some point in time, they just call it a polypeptide. And at some point in time, and this didn't come through, but this is the word protein right here. Roughly, it's a biological macromolecule, roughly around 5,000 grams per mole, which is called a Dalton. So a lot of times they don't talk about grams per mole when they use molecular weights. So a lot of times they use the term Dalton, which, you know, no one ever told me whenever I was in biochemistry in college, and I was even a biochem major, they never specifically had said, hey, you know, all of a sudden you'd see that term just disappear. Instead of saying something was 21,000 grams per mole, they would just say it was 21 kDAs, because a Dalton, the abbreviation for Dalton is a D, capital D, little a, but no one had ever said, hey, that's a gram per mole. Maybe they just assumed that I would know, but nope, a Dalton is a gram per mole. So, you know, in gen chem and organic chemistry, a lot of times we talk about things in grams per mole. You can still do that in biochemistry, but don't be alarmed when you reading on your enzymes or something and they say, oh, it's a 50,000 Dalton or 50 kDa protein. It just means it's 50,000. If it's kDa, obviously it means kilodalton. It'd be 50,000 grams per mole. Okay. And so, on your own, we're just going to briefly talk a little bit about this. So there are certain, you know, small peptides that are not proteins, but they're just small peptides. Some of them are naturally occurring, some of them are not. So I just always like to point out a couple. There's aspartame, which many times, yes, there are people that are allergic to it. Small enough doses, it's fine. And they show that, you know, even in mice, you have to go really high doses. So if you drink your Diet Coke, it's not going to kill you unless you're allergic to it. Now, there are people who definitely cannot do it. If you suffer from phenylketonuria, you can't because it does have phenylalanine in it. But all this aspartame is L-aspartyl, L-phenylalanine methyl ester. And so this is really just aspartate that's connected normally to a phenylalanine. The only thing that makes it unnatural is the fact that instead of it ending with an O-, it has an ester on the end, just a methyl group. Okay, and so, you know, it's naturally, your body perceives it as being sweet, but your body doesn't digest it as a sugar. So that's the reason why, you know, diabetics can use it. But once again, please use so in moderation, or if you're allergic to it, then don't use aspartame. All right, and then this is just another example of one. This is beta-alanine L-histidine, which is also commonly called carnosine. And I wanted to point this out in the sense that this, remember we talked about naturally occurring amino acids, which this is a naturally occurring compound, but the big difference here is this is not alpha. This is not an alpha amino acid. This is a beta amino acid. So it's got, this is the normally in alanine, you know, you'd have your methyl group here, but it's one removed. And so that's why it says beta-alanine. And if you remember from organic chemistry, the YL just means that it's a substituent. So it's beta-alanine L-histidine. Okay. And then, I don't know if the link still works or not in the original file, but it's actually been used, had some success at least, in using it as a dietary supplement in some forms of autism. And they don't, at least at the time that came out, they didn't know the reason why. They were hypothesizing, but they didn't know the reason why. I always like to point this one out, because if you look at these two, these are both hormones. One's oxytocin and one's vasopressin. Okay, and so we look, the only difference, what's one difference between oxytocin and vasopressin? One has what? One's got leucine, and one has arginine. What else? There are just two differences. This one, and that is isoleucine, and this one is phenylalanine. Typically, this one's considered a relatively minor modification because they're both hydrophobic. And so, yes, it's true that this one's aromatic and this one's branched, but they're both hydrophobic. The big difference here is the fact that you go from leucine, which what's the chemical property? What kind of amino acid is leucine? Non-polar, so it's going to be hydrophobic, hydrophilic. It's hydrophobic. To arginine, and arginine is polar. In fact, what is arginine? Not only is it polar, but it's hydrophilic, and it's an extreme. What's extreme about it? It's one of only three that, there's something else that is, what kind of amino acid? It's basic. It's positively charged under many things, under many conditions. Okay, what does oxytocin do? What is oxytocin? Pardon? Okay, and so what does it do though? What's it involved in? Besides giving an oxytocin high? Come on, at this point in time, you should have had it in multiple biology classes. Who has typically has higher concentrations of oxytocin? Men or women? Women. Okay, and what does it what men do have oxytocin. Now, if it gets too out of whack, it can be bad. But what's one of the functions that it has in women? Pardon? Trust me, and when this happens, you're just gonna wish you could have some oxytocin. During labor, it's what helps with the contraction for during labor. It also helps with the production of milk. Supposedly, it helps with the bonding, postpartum with the child and everything as well. What about vasopressin? It is not. It does not have to do with.
Something to do with, is it dilation or constriction?
With what did you say?
Is it something with constriction?
It can. It acts as an antidiuretic. Okay. And so it helps with the blood pressure, so it has to do with the uptake of water, release of water. Okay. So just a small change can have a very drastic difference between, giving birth and increasing your blood pressure. All right, and then I'm actually not going to go into a lot of detail on this one just because we don't have a lot of time. But phenylketonuria, I mentioned it already, and nowadays most people now don't have sodas in here. I used to always have people ask if they had like a diet soda or something in there, and there would be always this little warning if it was a diet drink, because it'll say, caution phenylketonurics, contains phenylalanine. It's because it has aspartame in it. So phenylketonuria is a disorder. It's a genetic disorder where we actually go over this kind of thing a lot. This is the kind of stuff that we discuss in Biochem 2, metabolic pathways. And this is the metabolic pathway that has to do with the degradation of amino acids. And what happens here is that since it's missing or it has a bad enzyme here, the phenylalanine hydroxylase, which converts phenylalanine to tyrosine, your body has to find a way to get rid of phenylalanine, and it goes using aminotransferase. And so it loses the amino group to make phenylpyruvate, which is a ketone. And this in of itself, which got blocked off, goes on to make, it gives off a carbon dioxide to make this compound. Which is called phenylacetate, okay? Which you should have remembered how to name that from organic chemistry, but that's phenylacetate. Phenylacetate is bad. It's very bad for you. Okay, and so this messes up the osmotic pressure in your brain, and you can die. You can die from it. So this is something that they routinely test for whenever a child is born in, at least in developed countries. Because once you're born and they know that you have this defect, PKU, then you have to go on a special diet your entire life. Many people with PKU have at least mild retardation or other defects just because it's hard to completely avoid it. But yeah, you're not supposed to take anything that has phenylalanine in it. So you have to be careful with your protein intake. All right, let me see where we at now. Is there something that we can kind of skip over or not hit? No, we really need to go over that part really well. All right, so this chapter is also the one that deals with protein purification. Like what happens if you're wanting to study a protein or isolate a protein in vivo? Okay, so you can imagine inside the cell there are billions of proteins out there, all different types. You're trying to find a specific one. So we have to talk a little bit ways to purify the protein. Part of it's going to depend upon the physical properties of the protein, and part of it is also on what part of the cell that it's in. Okay, so this is why they're just going to talk about here the fact that there are different ways to try to first isolate that protein from all the others, okay? And actually, you have to get all the proteins out of the cell, and there are some cool ways to do that. Okay? So they use the word, and if you're reading papers that have to do with proteins, they'll say homogenize or homogenization. And all that means is that you have to break open the cells in order to get the protein out, regardless of whether it's yeast or if it's mammalian or bacterial. So there are different ways. There are lots of different ways. I just picked a couple of them here. One is called sonication. Then there's also freeze thaw. And then sometimes you have to add detergents, and we'll talk about what that means. But then there are also ways called a French press. There's a French press. And there's one that's fun because it's called a homogenizer. So these are just some of the examples. This is just to show you, in rudimentary wise, you can use just like a mortar and pestle or something to literally smash the cells open. In fact, there's one way that's called a bead beater, which is where they literally take their sample of protein and they stick it into like a tube that has a bunch of glass beads they use glass because it's inert. You don't have to worry about reacting with something. And you close it and it just shakes it back and forth really, really hard, so like a paint mixer. And it literally just beats the cells open. Okay, and then you have to spin it down. We'll talk about that in a moment. But that's kind of crude. And you have to be careful because, crude because like literally not as in being, grotesque. Well, but you have to be careful because it gets hot. Anytime you break cells open, you think back to physics, it's going to be releasing heat. Okay, so heat will be released. You can also blend it sometimes. Especially like plants and stuff, a lot of times I'll initially blend it. Or if you're just using meat, like one time, for example, this is not the most legitimate way, but one of the research projects at the lab that I was working in, we were working at trying to get mitochondrial RNase-P. And so one part of your body that has a lot of mitochondria in it is a heart muscle, because it has to have so much energy, so it's going to have a lot more mitochondria than just other body parts. And obviously you can't very well just get a heart from a human. And so you can order hearts and order body parts, but first of all, they're expensive. And secondly, there's all this paperwork and everything, at least back in Michigan. And so what we would do is one of us would go to a slaughterhouse and we'd take our own bucket. And whenever they would slaughter the cow, we would pay a guy at the door and he would give us hearts. And so we found out he charged most of us $20, but he liked one guy, so we'd always send him because he would only get charged $10 per bucket of hearts. That way we didn't have to fill out any paperwork. We just literally slipped him a $10 bill or a $20 bill and get our bucket of hearts. But then what would happen was we get the heart back and we had to put it in a blender, the old wearing blender kind of thing, and blend it up first before you can do other things. All right. Now, one thing that we do have in the lab here, which is really cool, that I use as an undergrad and as a graduate student as well, and that Nolan used it this last semester. It's called a sonicator. And what does a sonicator use in order to break open the cells? Sound waves. And so what you do, this one is thick down here, but you literally stick this into your sample solution and it gives off high frequency sound waves and it bursts the cells open. It's much quicker, but you still have to worry about heat given off because as the cells break open, so you usually have to do it in ice, and it's actually very exothermic. Okay, and so you just turn up the frequency, you do a few cycles with sound, you burst it open, and so on and so forth. Pardon?
It sounds kind of like a weapon.
Yes. Mm-hmm. Yeah, that would be a horrible weapon to have because it would literally burst your cells open. And so, yeah, it sounds like something from a comic book. All right. So then another way, which I don't have a picture of, is a French press, which is just like, it's it's the same principle, except I don't know which came first. But the French press was and I don't have it, but it's a metal cylinder, like a piston, and it had a little tiny hole, just one little tiny hole at the very bottom. And then what you did is you took this piston and you forced it down, so you forced everything through this tiny little hole. And so by squeezing all the E. coli or the yeast through that hole, it burst open. But that one was more time consuming, plus the hole gets clogged up really easily. I hated using a French press. And so you have to do multiple passes. Whereas the homogenizer, it's really cool because, oh, wait a minute. It was this machine. And it was this big box. And it had a pump here, which was which you would have your solution and it'd be pumping it. And it had a pump on the way out. So you put it in another beaker on ice once again. Inside, it looked like a little maze or a little labyrinth. And so there are all these turns and twists and things like that. And what it would do is it would pump it through really, really fast. But there were so many, like the little labyrinth, the little maze, that the E. coli or the yeast would come and slam against the walls or slam into each other. Sort of like, what is that famous store in New York on wedding day for the brides? Surely you've seen that because it's been on multiple shows and stuff. When they had like the really basement bargain wedding dresses, and like these women will literally run over each other to try to get their wedding dress and stuff. Or like, Black Friday, just go to Walmart on, midnight of Thanksgiving night and you watch them. It's like homogenizer. And so usually this is really fast. So usually 1 or 2 passes because it's more effective than the French press because they're banging into each other throughout the entire labyrinth. But once you bang it open, you have to make sure that you separate the protein out from all the other crap that's in the cell. And that's the scientific term. So you use centrifugation, which just means you're spinning it really, really fast. Okay, because different organelles will spin out at different speeds. Okay, just spinning it at relatively low speeds, you can actually get rid of cell membranes and things. And sometimes your protein is in the cell membrane, and we'll talk about how to get it out of a membrane after a while. But sometimes, and I wouldn't expect you to know all of the different speeds and all this kind of stuff. But first of all, what they're showing here, this is a mortar and pestle, which is very similar to the idea as the French press, where you're just literally forcing. It's crude too, doesn't work very well, but it's a hard pestle, and you got it in a test tube, and you just keep like churning it up and down, and it breaks it open too by forcing it through the little holes. But then you have to spin it. If you spin it at relatively low speeds, the bigger things are going to come out, okay? You spin it at a higher speed, then you're going to get the mitochondria that will come out. And then ultimately, if you spin it the highest speed, which is showing, got cut off here, it's like 100,000 Gs, then you're going to get some of the other fine particulates like the ribosomes and stuff like that. And so your protein may come out at different parts. Or if you're lucky enough and it's in the cytosol, you can spin it at a really high speed, get rid of everything, and it's just left behind in all liquid, which that's ideal if you're just left behind in the cytosol and not part of the ribosome or part of the mitochondria or something. Otherwise, you have to do this fractionated stochastic method where you spin it at different speeds. Or what happens if, which actually isn't on here, you can imagine though, is if your protein, this is the phospholipid bilayer, it's just crudely drawn. And of course this is all negatively charged. We're going to find out later on. Everything's not completely negatively charged, but overall it has a negative charge. Okay, and let's say that you have your protein in here. There's this protein and you're wanting to get it out. If you try to just break open the cells, it's still going to get stuck in the membrane, right? Because it's hydrophobic. Obviously, it's hydrophobic. It likes to be in the membrane. So the way to get that out is you have to add detergent, which would make sense because that's what soaps do, right? Is what it's going to do is the soap is going to compete with this in order to hopefully get your protein to come out. And then when your protein comes out, you're going to have your protein and it's going to be surrounded by, the soap, whatever the soap detergent was. Okay, then you just have to try to separate the detergent off of it. So you know where your protein is located. One of the really, really nice things about proteins is that tyrosine and tryptophan are spectroscopically visible. So at the absorbance of 280 nanometers, DNA and RNA absorbs at 260. Tyrosine and tryptophan absorbs at 280. So you can see how much protein you have, or where protein is at, by looking at the 280 nanometers. There are also other spectroscopic, which is also called spectrophotometric, that's the same thing, methods. The most common one is called Bradford. I'm sure you probably use it in genetics or biology, where you just use a dye. It's mostly blue dye. It's similar to like blue ink. And what happens is it just binds the proteins, and then it absorbs at, I forgot, I want to say 600, but I may be wrong, but it absorbs at a different wavelength. Okay, and so you can actually get an idea of how much protein roughly that you have based off of how well it absorbs. Okay? Because the more protein it is that you have, the bluer it is. There are fine limitations, and next semester, if you take the lab, we actually will do some type of Bradford assay and UV vis, the one to look for tyrosine and tryptophan. We'll do that in lab. But use spectroscopy, and then UV vis, ultraviolet visible region of the spectrum. Okay, so we've been able to break open our cells. We've seen that we've got protein, but now we want to try to separate one specific protein from everything else. So the way to do that is you're going to have to take advantage of the physical properties of the protein. Okay? And so one way to do that is using column chromatography. And this goes all the way back. We actually did this in organic chemistry, and we even used the column. We weren't just doing proteins. Remember, we did dyes, like blue dye versus yellow dye and things like that. But it's very similar. I mean, it's identical. Same principle. So in your column, and don't hate me for my beautiful artistry, you're going to have some type of stationary phase. It could be beads, it could be some type of gel material, or something like that. And the stationary phase just does not move. So that's the reason why it's called stationary. Okay? Then what happens is you're going to have some type of mobile phase. So many times we're using something that has a water phase. So in biochemistry, usually, almost always, it's going to be a stationary solid phase and some type of liquid mobile phase. Now in organic chemistry, you can use, like, sometimes the phases are gas, like a gas chromatography unit. But here we don't typically use gas chromatography because you'd have to heat up the proteins to the point, we've never used gas chromatography to separate proteins out because you have to heat them up to the point where they'd fall apart. And so then what happens is, as you add your compound, your mixture there, based off of how well it liked the stationary phase, something that liked the stationary phase will get caught up in it and will take longer. Those that didn't like it's going to go right on through with the mobile phase. The mobile phase is called the eluent. Okay, just in case I ever say that term. So this is just a little picture which also got cut off. And so, but you can see here, they've added their sample, which is this darker color, and then based off of whatever the physical or chemical properties are of this mobile phase, is the different components of the mixture will start to separate. Okay, so then we have size exclusion or gel filtration, which sometimes people get these mixed up, but these are really cool. So what happens here? Well, that's really cool that I can do. Sorry. This is easier for me to redraw this. Size exclusion is usually the exact opposite of what you think it is. So it is going to separate proteins based off of the size, not based off of some chemical property. It doesn't care if it's positive or negatively charged, assuming it doesn't, precipitate out of the solution. But what happens here instead is there are these beads. And these beads kind of look like Swiss cheese. You can get them that have different size holes through them. Of course, there are these holes all over the place. Then what happens is as you add your mixture, what do you think is going to come off first? Well, I mean, kind of it already tells you. But what happens is if we have a protein that's relatively large, it can't fit in those little holes, and so it just goes between the big beads. So that's what's called size exclusion. Those sizes, they're excluded from going inside. Whereas if you have something that's small enough that can go inside, it's going to go through all the little different types of holes, and it takes a lot longer for it to come out. So it separates it based off of size, but the larger items come out first. The smaller items come out last. Now you don't want to confuse this with like regular gel electrophoresis, which is the exact opposite. Because in those kind of columns, which you can run a column with that material, what it's going to look like is this. There, instead of having, you can still have beads, but usually it's some type of matrix. And what happens here is it's uniform. It's not like different size holes. And so what happens is our small things can wiggle through, whereas the big things get caught, and it takes a lot longer to work its way through the column.
Oh, so the large comes out.
So if it's like a gel material,
Oh,
then yeah. Whereas if it's size exclusion, it's the exact opposite. Because, okay, so that's why a lot of times when you probably in genetics ran DNA or protein gels to where the bigger things took longer and the smaller things, because it was using something essentially more like gel affinity to where it was a matrix. Whereas you can do the exact opposite and do a size exclusion where the smaller things take a lot longer because the big things just go right on through. Okay.
So size exclusion is like the key term for defining the difference between the two?
Yes, sometimes you hear them used interchangeably, but typically speaking, size exclusion means that the small things are going to take longer. Okay. So I usually try not to say just gel affinity because then that's kind of tricky because it also depends what the gel is. So I usually will say like gel electrophoresis or that if we're using polyacrylamide, which is literally just a polymer, then it's the one where the big things will get caught, get stuck, and the small things go through. Whereas if it says size exclusion, it's the exact opposite. Okay. And this is where, well, actually we have just a couple minutes left. I just wanted to go right on through to skip ahead. To affinity chromatography and just talk about it briefly, and this really didn't come through at all. So affinity chromatography is actually really simple. One of the more simple ones to think about is, once again, you have beads or something, but there's some type of chemical property on that bead that proteins will have an affinity for. Okay, and so if they like it, they're going to stay on the column. If they don't like it, they're going to go right on through. And so you could have different types of affinity. One could be like hydrophilic or hydrophobic. You can fill up the column with something that's hydrophobic, then the hydrophilic proteins are going to go right on through, and the hydrophobic ones will get stuck. Okay, or you could do the exact opposite. You can have them charged. You can have ones positively charged, so then the proteins that are more negatively charged will stay on there, and the ones that are positively charged will go on through, or vice versa. And so on the next class, we'll pick up with this just a little bit, but I would like to then really get started with chapter 4, which really picks up right where chapter 3 leaves off. So I'll go ahead and I'll end